Hello friends, warm welcome in this video, that is epitopes. Friends, as you know that immune cells or lymphocytes do not interact with or recognize an entire immunogen molecule. Instead of that, lymphocytes recognize discrete site on the antigen molecule, and these discrete sites are called as epitopes or antigenic determinants. Thus, epitopes are the immunologically active regions of an immunogen that bind to antigen-specific membrane receptor on lymphocytes or to secreted antibodies. Studies have shown that B and T cell recognize different epitopes on the same antigenic molecule. For example, when mice were immunized with a small human hormone of 29 amino acids, that is glucagon, then antibody will be elicited to the amino terminal portion of this hormone, whereas the T cell will respond only to epitopes in the carboxyl terminal portion of this protein. Lymphocytes may interact with a complex antigen on several levels of antigen structures. An epitope on protein antigen may involve elements of primary, secondary, tertiary, and even quaternary structure of the protein. In polysaccharides, branched chains are commonly present, and multiple branches may contribute to the three-dimensional structure of the epitope. Lymphocytes may interact with a complex antigen on several levels of antigen structure. An epitope on protein antigen may involve elements of the primary, secondary, tertiary, and even quaternary structure of the protein. In polysaccharides, branch chains are commonly present, and multiple branches may contribute to the three-dimensional structure of the epitope. The recognition of antigen by T cell and B cell is fundamentally different, as the B cell bind antigen that are free in solution. The epitopes they recognize tend to be highly accessible sites on the exposed surface of the immunogen. On the contrary, most T cells recognize only peptides combined with MHC molecules on the surface of antigen presenting cells and altered self cells. T cell epitopes, as a rule, cannot be considered apart from their associated MHC molecules. So now we will learn here various properties of B cell epitopes. Several generalizations have emerged from studies in which the molecular features of the epitopes recognized by B cell have been established. The ability to function as a B cell epitopes is determined by nature of antigen binding site on the antibody molecule displayed by B cells. Antibody binds to an epitope by weak non-covalent interactions which operate only over short distances. For a strong bond, the antibody binding site and the epitope must have complementary shapes that place the interacting groups near to each other. This requirement poses some restriction on the properties of the epitope. The size of the epitope recognized by B cell cannot be larger than the size of antibodies binding site. For any given antigen-antibody reactions, the shape of the epitope that can be recognized by the antibody is determined by the shape assumed by the sequence of amino acids in binding site and the chemical environment that they produce. Smaller ligands such as carbohydrates, small oligonucleotides, peptides, and heptanes often bind with a deep pocket on antibody. For example, angiotensin II, which is a small octopeptide hormone, binds with a deep and narrow groove of a monoclonal antibody specific for the hormone. Within this groove, the bound peptide hormone folds into a compact structure with two turns, which brings its amino terminal and carboxyl terminal close to each other. All 8 amino acids residue of the octopeptide are in van der Waals contact with the 14 residue of the antibody groove. A quite different picture of epitope structure emerges from X-ray crystallographic analysis of monoclonal antibodies bound to globular protein antigens such as hen egg white lysozyme and neuraminidase. Neuraminidase is a enveloped glycoprotein of influenza virus. These antibodies make contact with the antigen across large flat face. The interacting phase between antibodies and epitopes is a flat or undulating surface in which protrusions on the epitopes or antibody are matched by corresponding depressions on the antibody or epitopes. These studies have revealed that 15 to 20 amino acids on the surface of the antigen make contact with similar number of residues in the antibody's binding site. The surface area of this large complementary interface is between 650 angstrom and 900 angstrom square. For these globular protein antigens, then the shape of the epitope is entirely determined by tertiary conformation of the native protein. The B cell epitopes on native proteins generally are composed of hydrophilic amino acids on the protein surface that are topographically accessible to membrane-bound or free antibodies. A B cell epitope must be accessible in order to be able to bind to to an antibody. In general, protruding regions on the surface of the protein are the most likely to be recognized as epitopes, and these regions are usually composed of predominantly hydrophilic amino acids. Amino acid sequences that are hidden within the interior of a protein often consist of predominantly hydrophobic amino acids and cannot function as B-cell epitopes unless the protein is first denatured. B-cell epitopes can contain sequential or non-sequential amino acids. Epitopes may be composed of sequential contiguous residues along the polypeptide chain or non-sequential residues from segments of the chain brought together by the folded conformation of an antigen. Most antibodies elicited by globular proteins bind to the protein only when it is in native conformation. Because denaturation of such antigens usually changes the structure of their epitopes, antibodies to the native protein do not bind to the denatured protein. 5 distinct sequential epitopes, each containing 6 to 8 contiguous amino acids, have been found in sperm whale myoglobin. Each of these epitopes is on the surface of the molecule at bends between the alpha helical regions. The sperm whale myoglobin also contains several non-sequential epitopes or conformational determinants. The residues that constitute these epitopes are far apart in the primary amino acid sequence but close together in the tertiary structure of the molecule. Such epitopes depend on the native protein conformation for their topographical structure. One well-characterized non-sequential epitope is hen-egg-white lysozyme. Although the amino acid residues that compose of this epitope are far apart in the primary amino acid sequence, they are bound together by the tertiary folding of the protein. Sequential and non-sequential epitopes generally behave differently when a protein denatured, fragmented, or reduced. For example, appropriate fragmentation of sperm whale myoglobin can yield 5 fragments, each retaining one sequential epitope, as demonstrated by the observation that antibody can bind to each fragment. On the other hand, fragmentation of protein or reduction of disulfide bonds often destroys non-sequential epitopes. For example, hen-egg-white lysozyme has 4 intrachain disulfide bonds, which determines the final protein three-dimensional structure. Many antibodies to hen-egg-white lysozyme recognize several epitopes, and each of 8 different epitopes have been recognized by distinct antibody. Most of these epitopes are conformational determinants dependent on overall structure of the protein. If the intrachain disulfide bonds of hen-egg-white lysozyme are reduced with a mercaptoethanol, the non-sequential epitopes are lost. For this reason, antibody to native hen-egg-white lysozyme does not bind to reduced hen-egg-white lysozyme. B-cell epitopes tend to be located in flexible regions of immunogen and displays site mobility. Scientists analyze the epitopes on a number of protein antigens, that is myohemorrhine, insulin, cytochrome C, myoglobin, and hemoglobin. By comparing the positions of the known B-cell epitopes with the mobility of the same residues, their analysis showed that the major antigenic determinants, that is epitopes, in these proteins generally were located in the most mobile regions. These investigations proposed that site mobility of epitopes maximize complementarity with the antibody binding site, permitting an antibody to bind with an epitope that it might bind ineffectively if it were rigid. However, because of loss of entropy due to binding to a flexible site, the binding of antibody to a flexible epitope is generally of lower affinity than the binding of antibody to a rigid epitope. Friends, complex proteins contain multiple overlapping B-cell epitopes, some of which are immunodominant. For many years, it was considered that each globular protein had a small number of epitopes, each confined to a highly accessible region and determined by the overall three-dimensional structure of the protein. However, it has been shown more recently that most of the surface of globular protein is potentially antigenic. This has been demonstrated by coupling the antigen binding profiles of different monoclonal antibodies to various globular proteins. For example, when 64 different monoclonal antibodies of a bovine serum albumin, that is BSA, were compared for their ability to bind to a panel of 10 different mammalian albumins, 25 different overlapping antigen binding profiles emerged, suggesting that these 64 different antibodies recognized a minimum of 25 different epitopes on the bovine serum albumin, that is BSE. The surface of a protein presents a large number of potential antigenic sites. The subset of antigenic sites on a given protein that is recognized by immune system of an animal is much smaller than the potential antigenic repertoire, and it varies from species to species and even among individual members of a given species. Within an animal, certain epitopes of an antigen are recognized as immunogenic, but others are not. Furthermore, some epitopes called immunodominant induce a more pronounced immune response than other epitopes in a particular animal. It is highly likely that the intrinsic topographical properties of the epitopes as well as animals' regulatory mechanism influences the immunodominance of epitopes. So now we will learn the properties of T-cell epitopes. Antigen-derived peptides are the key elements of T-cell epitopes. Scientists suggested that there was a qualitative difference between the T-cell and the B-cell response to protein antigen. They compared the humoral, that is B-cell, and cell-mediated, that is T-cell responses to a series of native and denaturing protein antigens. They found that when a primary immunization was with a native protein, could elicit a secondary antibodies, that is humoral response, but not denatured protein. In contrast, both native and denatured proteins could elicit a secondary cell-mediated response. The findings that secondary response mediated by T-cell was induced by denatured protein even when a primary immunization had been with native protein. Now it has become clear that T-cells do not recognize soluble native antigens but rather recognize antigens that has been processed into antigenic peptides, which are presented in combination with MHC molecule. For this reason, destruction of the conformation of a protein by denaturation does not affect its T-cell epitopes. Because the T-cell receptors does not bind free peptides, experimental system for studying T-cell epitope must include antigen presenting cells or target cell that can display the peptides bound to an MHC molecule. Antigenic peptides recognized by T-cell form trimolecular complexes with a T-cell receptor and MHC molecule. The structure of T-cell receptor peptide MHC trimolecular complex have been shown here. Unlike B-cell epitopes, which can be viewed strictly in terms of their ability to interact with antibody, T-cell epitopes must be viewed in terms of their ability to interact with both T-cell receptor and MHC molecule. The binding of MHC molecule to an antigenic peptide does not have the fine specificity of interaction between an antibody and its epitope. Instead, a given MHC molecule can selectively bind a variety of different peptides. For example, the class 2nd MHC molecule bind peptides from ovalbumin, hemagglutinin, and lambda repressor. Studies reveals structural features or motifs common to different peptides that bind to a single MHC molecule. Antigen processing is required to generate peptide that interact specifically with a MHC molecule. Endogenous and exogenous antigens are usually processed by different intracellular pathways. Endogenous antigens are processed into peptides within the cytoplasm, while exogenous antigens are processed by endocytic pathway. Epitopes recognized by T cells are often internal. T cells tend to recognize internal peptides that are exposed by processing within antigen presenting cells or altered self cells. So friends, if you like this video, please subscribe to our channel, press the bell icon, and don't forget to share this video among your friends. Thank you.